Today we are going to talk about that when adrenergic receptors are stimulated, how they induce intracellular signals, right? What are the molecular basis of adrenergic receptors function, right? So let's take different tissues. Let's suppose here is your central nervous system, right? Preganglionic neuron and then postganglionic neuron releasing norepinephrine here and here, here and here. Let's suppose these are the nerve endings, and from here it is releasing norepinephrine. Now we have to see that how norepinephrine work on the adrenergic receptors. I will put one model here. I will explain in this diagram first how the alpha 1 adrenergic receptors work, right? Alpha 1 adrenergic receptors work. So this is the nerve ending which is going to release, yes, norepinephrine, right? Now, once norepinephrine is released, it will bind with the adrenergic receptors. All the adrenergic receptors are belonging to the family of 7-pass receptors, right? Why we call them 7-pass receptors? Because these receptors are peptide chain, and this peptide chain passes through the cell membrane 7 times. 1, 2, 3, 4, 5, 6 and 7. Now this pocket where norepinephrine binds, right? This pocket of the peptide chain, this red peptide chain is the adrenergic receptor. Let's suppose that this is alpha 1 adrenergic receptor, right? Now alpha 1 adrenergic receptor, like other adrenergic receptors, it is a 7-pass receptor. We also call it serpentine receptor. Serpentine means snake-like receptor, right? And intracellularly, these receptors couple with G proteins. So third name for such receptor is G protein coupled receptor. So what we have to remember that all adrenergic receptors are, yes, they have 3 names. They can be called 7-pass receptors because these receptors pass through the membrane 7 times. 7-pass receptors, they can also be called, yes, serpentine receptors, right, because they are like snake. And then they can also be called G proteins, yes, coupled receptors. Even receptors are sexy, you know. G protein coupled receptors, right? Now, alpha 1 adrenergic receptor, like other receptors, they must have one extracellular domain where norepinephrine binds. So norepinephrine binds over here, right? And this point where norepinephrine binds, this is called ligand binding domain. And this intracellular part of the receptor, this is called effector domain. This is called effector domain. Now this effector domain, in case of alpha 1 adrenergic receptor, when, right, effector domain become activated when norepinephrine bind with the receptor, and this activated effector domain will stimulate G protein, right? Now which G protein it stimulates? Which G protein it stimulates? GQ. It stimulates the GQ. G proteins are primarily, yes, G proteins are primarily trimeric proteins. They have alpha component, they are having beta component, and they are having gamma component, right? What really happens when this receptor is stimulated by epinephrine or norepinephrine, it will stimulate the alpha component of the G protein, and this G protein is called, yes, this is called GQ, and this alpha is called alpha Q. Now as soon as the receptor activate the alpha component of the G protein, alpha component which was previously binding GDP, it loses the GDP and binds GTP. GTP is high energy molecule, but it binds with the GQ alpha component, then alpha component dissociate with beta and gamma, right? Alpha component dissociate from beta and gamma. This dissociated alpha component, this is now alpha Q, and it is loaded with GTP, and now it is fully active, and this activated alpha component, and of course your friends beta and gamma have been dissociated. Beta and gamma are dissociated from this trimeric complex. Now alpha component will bind with, yes, there is an enzyme here. What is the name of this enzyme? Adenylyl cyclase, and this enzyme is very happy because you remember its name. Very good, right? So everyone is happy when you remember their name, including the enzyme. So adenylyl cyclase is the effector molecule on which, oh, you know, we are wrong. We are dead wrong, because it is alpha stimulatory which bind with adenylyl cyclase. Enzyme is happy because you are fooled. Yeah, GQ does not bind with adenylyl cyclase, rather GQ bind with a different enzyme, and that enzyme is called, yes, who will tell me the name of that enzyme? Yeah, this is the name of that enzyme. Let me tell you. Yeah, this enzyme can attack the cell membrane. This enzyme can attack the cell membrane. When this enzyme is activated, what is the name of this enzyme? Phospholipase C. So actually when GQ is stimulated, the alpha component of GQ stimulates the phospholipase C. And when phospholipase C is activated, this enzyme attacks the lipids in the cell membrane, right? This will attack the lipids in the cell membrane, a very special lipid which is in the cell membrane. This is called, yes, this lipid is called phosphatidyl inositol diphosphate. What is this lipid? Phosphatidyl inositol diphosphate, and this lipid will be attacked by the phospholipase C. When phospholipase C will attack the phosphatidyl inositol diphosphate, this will break down into two components, right? It will break down into two components. One is inositol, yes, triphosphate, IP3. Other is diacyl glycerol. Then you must be knowing that inositol triphosphate actually act on intracellular stores of calcium. Within the cell, calcium is stored in endoplasmic reticulum, right? So calcium is stored well in endoplasmic reticulum, and here is a calcium transporter, right? And calcium is well stored. What is this? Endoplasmic reticulum, which is very rich in calcium. And what really happens, that IP3 will stimulate this pump. IP3 will stimulate this pump. When this pump is stimulated by IP3, or exchanger is stimulated, calcium will come out, and intracytosolic calcium levels will go up, and this calcium will bind with special type of kinases right? Calcium will bind with colmodulin. You know the protein called colmodulin, right? And this protein to which calcium is binding, that is called colmodulin kinases. This protein is called colmodulin kinase, right? Calcium will bind with colmodulin kinase protein, right? Now, if you really want to know the steps, calcium will bind with colmodulin, and calcium colmodulin complex will activate certain kinases which are called CAM kinases. Now, once calcium colmodulin kinases or CAM kinases are activated, this group of enzymes will phosphorylate target protein. For example, one example of target protein in smooth muscle is, yes, you know, so alpha 1 adrenergic receptor produce smooth muscle contraction. You remember that? Very good. So how it will do? Calcium colmodulin will phosphorylate light chain kinases. There is an enzyme called, yes, light chain kinases, right? And when these light chain kinases are acted upon by calcium CAM kinases, then light chain kinases are phosphorylated. They become active. They become phosphorylated because kinase enzyme add phosphate to light chain kinases. How? They take the ATP and break the ATP into ADP, and phosphate is added to this. When light chain kinases are phosphorylated, who will tell me the next step? They will phosphorylate myosin light chains. They will lead to phosphorylation of myosin light chains phosphorylated. When they become phosphorylated, right, the phosphorylated myosin light chains can participate in actin-myosin interaction. What will happen? Myosin light chains which are phosphorylated, they can participate in actin-myosin interaction. And this actin-myosin interaction will lead to, yes, smooth muscle contraction. So this is one example how alpha 1 adrenergic receptors work. That alpha 1 adrenergic receptors, once they are binding with the ligand, they stimulate the GQ protein. The alpha component of GQ stimulate phospholipase C enzymes which break down the intramembranous phosphatidyl inositol diphosphate into IP3 and diacylglycerol. What IP3 is doing? IP3 can act on certain calcium exchangers on the endoplasmic reticulum and release the sequestered and calcium right from the endoplasmic reticulum into cytosol. Calcium will bind with the colmodulin, and calcium colmodulin kinases will be activated, which will phosphorylate target protein. In smooth muscle, target protein is light chain kinases. When light chain kinases become phosphorylated, they phosphorylate myosin light chains. When myosin light chains are phosphorylated, yes, now they are very happily interacting with the actin. So actin myosin interaction start and smooth muscle will contract. Am I clear? Secondly, don't forget this DAG, diacylglycerol. Diacylglycerol is another second mediator produced by phosphatidyl inositol diphosphate, and diacylglycerol can activate. Who will tell me? Diacylglycerol can activate? Yes, please. Another kinase. What is that? Protein. Yes, protein kinase C. Right? So protein kinase C will be phosphorylated. And when protein kinase C is phosphorylated, right, or activated, diacylglycerol activate, listen again, diacylglycerol actually, let me repeat it again, diacylglycerol will act on protein kinase C, and protein kinase C will become, yes, C will become activated. No, please correct, it is not phosphorylated, it becomes simply activated. When protein kinase C is activated in the presence of diacylglycerol, then protein kinase C can phosphorylate certain target proteins, right? Target proteins phosphorylated. So what did we really learn? We learned that whenever norepinephrine or epinephrine or any other compound which is having agonist action on alpha-1 adrenergic receptor, right, that will lead to activation of intracellular GQ protein, activation of phospholipase C, increase intracellular level of IP3 and diacylglycerol, and IP3 will increase intracytosolic calcium levels. Calcium will bind with calcium calmodulin kinases, and diacylglycerol will bind with protein kinase C. When both of these kinases are activated, they will phosphorylate the target proteins, and phosphorylated target proteins will alter the function of the cell. Am I clear? Any problem here? So this was how alpha-1 adrenergic receptors work. If someone asks a very simple question, that when alpha-1 adrenergic receptors are stimulated, which intracellular second messenger's concentration is increased? Then Dr. Nikita will answer, only she will answer, which intracellular second messenger level will increase? What is the second messenger here? Yes? Okay, IP3 and diacylglycerol, but more importantly, calcium. Is that right? Intracellular calcium will increase, right? Now we come to action of, we have studied the action of alpha-1 adrenergic receptor. Now we will study the action of alpha, yes, 2 adrenergic receptor, how alpha-2 adrenergic receptors work, right? Let's suppose this is alpha-2 adrenergic receptor. Again, you can find alpha-2 adrenergic receptor is also 7-pass receptor, or other name is serpentine receptor, or it is G protein coupled receptor. And whenever norepinephrine or epinephrine binds here and alpha-2 adrenergic receptor is stimulated, intracellularly it will stimulate not GQ. Alpha-1 was coupled with GQ. Alpha-2 receptor is not coupled with GQ, it is coupled with, yes please, G inhibitory. This is coupled with G inhibitory protein, and G inhibitory protein is alpha inhibitory, and with that it has beta and, I think I should change the color so that you don't get confused, right? So G inhibitory has alpha inhibitory, with that it is having, yes, beta inhibitory and gamma inhibitory. So whenever alpha-2 receptor is stimulated, alpha inhibitory component of the G protein will lose the, yes, GDP and acquire the GTP, and now it will become energized and it will dissociate from its friends, right? Alpha inhibitory, which is energized, now it is activated, right? Because it is loaded with, yes, GTP. And of course, when alpha inhibitory will dissociate from beta and gamma, as alpha inhibitory has separated, in the same way beta gamma units will be also separated, beta gamma inhibitory units. Then what will happen, that alpha inhibitory will interact with your friend, yes, you remember now, adenylyl cyclase, and now, yeah, what will happen to adenylyl cyclase? It will bind to this component of the adenylyl cyclase, and whenever adenylyl cyclase has two domains, at one domain alpha inhibitory bind, it has another domain, and with this domain, this is a different domain. With this domain, alpha stimulatory bind. So alpha inhibitory binds to this domain, and whenever alpha inhibitory bind, it will lead to inhibition of action of adenylyl cyclase. And when adenylyl cyclase is inhibited, then ATP cannot be converted into cyclic AMP. So G inhibitory or alpha inhibitory component of G inhibitory inhibit adenylyl cyclase. So conversion of ATP into cyclic AMP is reduced. When there is reduced cyclic AMP, so of course then there is reduced cyclic AMP activate, yes, protein kinase A. So there is reduced activation of protein kinase A. Then protein kinase normally phosphorylate target protein. But now because protein kinase A is not stimulated because cyclic AMP levels are less intracellularly, so protein kinase A dependent intracellular activities are reduced. So what should I write? When protein kinase A is not activated, then phosphorylation of protein kinase A proteins don't occur. And we say that there is reduced activity of protein kinase A dependent, yes, intracellular functions. For example, if in this cell there was an enzyme which was acted upon by protein kinase A, and when that enzyme was getting phosphorylated, a metabolic pathway was run. But when alpha 2 receptor is stimulated, G inhibitory is activated, and activated G inhibitory inhibit adenylyl cyclase, cyclic AMP level become less, protein kinase A activity become less, then that enzyme will not be phosphorylated, and that enzyme dependent metabolic pathway will be inhibited. Am I clear? Secondly, a very important factor which you must know is beta inhibitory and gamma inhibitory component. Once they dissociate from alpha inhibitory, they can bind with the potassium channels. They are special potassium channels, and these potassium channels have intracellular component where beta inhibitory and gamma inhibitory can bind. So this beta and gamma inhibitory bind with the, what is this? Potassium channel. And as you know, the potassium concentration intracellularly is high. So naturally potassium will start moving from intracellular compartment to extracellular compartment. So what really happens, beta gamma unit of G inhibitory increase potassium efflux. So when the cell is losing the potassium, naturally what will happen to this situation? Yes, cell will become electrically more negative because cell is losing positive charges. So we can say if this cell was having resting membrane potential of suppose minus 70 millivolt, but once potassium is lost, then resting membrane potential will become more negative because positive charges are lost. So it will become minus suppose 90 millivolt. So what really happens, the stimulation of alpha 2 adrenergic receptor, right, which when it activates the G inhibitory, activated beta and gamma units of G inhibitory activate the potassium channels and let the potassium be lost from the cells, and when intracellular potassium is lost outside, so resting membrane potential become more electronegative. It means cell become more polarized towards the negative side, or we simply say cells become hyperpolarized and they become electrically inhibited. Am I clear to everyone? Another point, this is what very few very good students know only, that alpha adrenergic receptors have one more function. This is alpha 2 adrenergic receptor. When ligand binds with it, not only these receptors activate G inhibitory, they also activate a very special type of G which is called G naught, alpha naught and beta gamma units. This is a different type of G protein. This is called G naught. You must be knowing GQ, you must be knowing G inhibitory, we'll study about G stimulatory, and this is a different which is called G naught. Actually, alpha 2 adrenergic receptors can stimulate the G naught also, and when G naught is stimulated, of course alpha component will lose the GDP and acquire the GTP, and alpha component become activated. And when alpha component becomes activated, then alpha naught loaded with, yes, GTP, right? This binds with the calcium channels. It binds with the, yes, which channels? Calcium channels. And when this will, alpha naught will bind with the calcium channel, it will inhibit the calcium channel. So calcium influx will stop. Calcium influx will stop. This is what happens in the presynaptic nerve endings. If you remember that in the very early lectures of adrenergic system, if this is one neuron and this neuron is releasing norepinephrine here, right, and this is your target tissue, I told you norepinephrine work on presynaptic membrane, right, through which receptors? Through, yes, alpha 2 adrenergic receptor, and I mentioned that when norepinephrine act on presynaptic membrane, inhibit the nerve ending functions. How it inhibit? Number one, it will, when G inhibitor is stimulated, activated, it activates the potassium channels. And when this nerve ending will lose the potassium, it will become hyperpolarized, it become very negative, so this will be difficult to stimulate this nerve ending. This is one change. Secondly, G naught will block the calcium influx, and when calcium influx is blocked to this nerve ending, the exocytosis of the neurotransmitter release will be less. Thirdly, G inhibitory, right, will inhibit adenylyl cyclase, and intracellular protein kinases, and at this point will be inhibited, and they will inhibit the enzymes and transporters which are concerned with synthesis and storage of norepinephrine. So these are multiple mechanisms how presynaptic alpha 2 adrenergic receptors inhibit the presynaptic membrane. Is that right? No problem up to this? Now we come to we have learned what are the signaling mechanism of alpha 1 adrenergic receptor. We have learned what are the signaling mechanisms in alpha or molecular mechanism in alpha 2 adrenergic receptors, and now we'll talk about what are the molecular basis of signaling mechanism of beta 1 adrenergic receptors and beta 2 adrenergic receptors and beta 3 adrenergic receptors. Again, there is a question. Let's have a break. Now we will talk about the intracellular signaling mechanism for beta receptors, right? Beta 1, beta 2 and beta 3. The good news is that the intracellular signaling mechanism for beta 1, beta 2 and beta 3 are same, right? So let's study the beta 1 intracellular mechanism. When beta 1 adrenergic receptor is stimulated, let's suppose this is beta 1 adrenergic receptor, right, and if epinephrine or norepinephrine, right, with beta 1, let's suppose epinephrine binds here, right, epinephrine. This is the beta 1 adrenergic receptor. You remember alpha 1 adrenergic receptor was coupled with GQ. Alpha 2 adrenergic receptor was coupled with G inhibitory, but beta 1 adrenergic receptor is coupled with G stimulatory, right? It is coupled with G stimulatory protein. So let's suppose this is alpha component of alpha stimulatory, this is beta and gamma stimulatory, right? When epinephrine bind with beta 1 adrenergic receptor, it intracellularly stimulate the G stimulatory. Which component of the G stimulatory? alpha component of the G stimulatory, and then of course alpha component on stimulation loses GDP and acquires GTP, right? And this alpha component also dissociate from beta and gamma. This alpha stimulatory component which is loaded with GTP, right? It dissociates from beta and gamma component, right? And it interact with, yes please, with adenylyl cyclase. If you remember, adenylyl cyclase has 2 points. There was one point where G inhibitory could bind, other point where alpha component of G stimulatory could bind. And when, right, this adenylyl cyclase is stimulated by alpha stimulatory, right, then what really happens? That this will stimulate the activity of adenylyl cyclase, and adenylyl cyclase will start converting ATP into cyclic AMP. So there is raised intracellular cyclic AMP level. Again, when beta 1 adrenergic receptors are stimulated, they are coupled with intracellular G stimulatory protein, and G stimulatory protein alpha component stimulate adenylyl cyclase, which convert more ATP into more cyclic AMP. And this cyclic AMP is the second messenger of this system. This second messenger molecule will stimulate, yes, cyclic AMP molecule will stimulate, yeah, what is this? Protein kinase A, right? And this protein kinase A will start phosphorylating the target protein. It will start phosphorylating target proteins, right? Whatever target protein is there, right, that protein will be phosphorylated. Now, when proteins get phosphorylated, their functions are altered. In this way, a target cell's functions will be altered. As you know, now let me give you a few examples. Beta 1 receptors, when they are stimulated, there is cardiac stimulation, right? There is cardiac stimulation. There is increased heart rate. There is increased contractility. Now, how it really happens? It happens by 2 ways. The one of the target protein may be calcium channel. Here is the calcium channel. And if protein kinase A phosphorylate the calcium channel, and when calcium channel get phosphorylated, it work more efficiently, and there will be more calcium influx. There will be more calcium influx, and calcium dependent intracellular mechanisms will be accelerated. For example, when calcium will, again, protein kinase A, when it is stimulated, it can phosphorylate calcium channels. Suppose these calcium channels are present in SA node, then there will be more calcium influx in SA node. And you know that in SA node and AV node, action potential is dependent on calcium, right? In the same way, calcium influx will occur in atrial and myocardial cells also. And myocardial cells, contractility is also dependent on calcium, so there will be positive inotropic action, right? So, but one thing which many students don't know, that alpha stimulatory can directly, now this is a very important point, alpha stimulatory can directly stimulate, alpha stimulatory can directly bind with the calcium channels and stimulate it directly also, right? So in this way we can say that when G stimulatory proteins are stimulated, right, by the stimulation of beta 1 receptors, right, intracellular cyclic AMP level increase, protein kinase A activity increase, and protein kinase A will phosphorylate target proteins and bring changes in intracellular function. Plus, what we have to talk about, that alpha stimulatory can directly bind with the calcium channels and increase the activity of calcium channels, so calcium influx becomes more, and calcium dependent electrical activity in the myocardium and calcium dependent contractile activity of the myocardium will be amplified. Am I clear? As I told you, the good news is that beta 1 receptors, beta 2 adrenergic receptors and beta 3 adrenergic receptors, all of them are coupled with intracellular G proteins, and in all of them there is increased intracellular cyclic AMP level, right? So the intracellular signaling mechanism for beta 1 and beta 2 and beta 3 is the same, right? Now let's do a little review and then we'll continue further. Let me put here adrenergic receptors. Right, already you know adrenergic receptors are divided into alpha and yes, beta. Alpha are divided into alpha 1 adrenergic receptors, alpha 2 adrenergic receptors, beta are divided into beta 1 adrenergic receptors, beta 2 adrenergic receptors and beta 3 adrenergic receptors. Now these adrenergic receptors, which is alpha 1 adrenergic receptors, they are coupled intracellularly with which type of G protein? Just please. GQ. And what is the second messenger system? Yes please. IP3 diacyl glycerol, leading to and eventually leading to increased intracellular calcium level. Excellent. Now we'll talk about alpha 2 adrenergic receptors. Right, alpha 2 adrenergic receptors, they are intracellularly coupled with which G protein? G inhibitory, and that will lead to, yes please, decreased, that will lead to actually three types of activity. G inhibitory, when it is stimulated, number 1, yes, decreased intracellular cyclic AMP. Number 2, there is increased potassium efflux, and number 3, there is decreased, decreased calcium influx. Am I clear? No problem. Now we come to beta 1 adrenergic receptors. Beta 1 adrenergic receptors are intracellularly coupled with, yes please, G stimulatory. And what will be the result in the cell? There is, yes, increased intracellular, yes, cyclic AMP level. Plus it has one more thing. G stimulatory has two activities. One is this and other is, yes, increase calcium influx. Increased calcium influx. And then there is the good news that beta 2 receptor mechanism and beta 3 receptor mechanism is the same as beta 1. Any question up to this? There is no question. That's it. Yes?
The only one of these receptors, which is the one that the beta and the you know that it stimulates the alpha portions?
Yeah.
And the beta and the gamma portion, which is the one that stimulates the potassium channel?
Which component stimulates the potassium channel? Potassium channel is stimulated by beta gamma unit of the G inhibitory, right? Okay. And calcium channels are blocked by the G naught, right? And here calcium channels are opened by the G stimulatory directly, alpha stimulatory, and also by the phosphorylation of the calcium channels. Any more question? No. Okay, so now we go to the next area. Next area is really very important and applies to lot of areas in pharmacology and endocrinology and neurotransmissions in central nervous system and peripheral nervous system.